Hi there. Now that we've gained the ability to look at 22,000 genes and get information about their expression levels, we're confronted with a problem of data overload. In this video, we'll talk about some of the approaches, computational approaches, that are used to analyze and try to make sense of this large amount of data. We'll talk mostly about the technique of clustering, which is going to try to identify genes that behave similarly and conditions in which appear similar molecularly. We'll focus on hierarchical clustering, although there are many different clustering methods, and then we'll also talk about enrichment analysis to identify genes that might be statistically overrepresented in a sample. So this slide reminds us that regardless of format, spotted microarray or Affymetrix, we're going to have two fluorescent values. We will compute a ratio in terms of the fluorescence intensity, or with Affymetrix, we have two individual chips, two individual samples. We have fluorescence values for each, and we will export these fluorescence values into a matrix or Excel spreadsheet often with a list of genes, a list of conditions, and a value for the ratio or the total intensity. But the numbers themselves don't tell much of a story, at least to a human being. And so one thing that's going to try to be done is we're going to try to find things that behave similarly. And what we have here is a heat map representation of the Excel spreadsheet from the previous slide. We have a list of genes, maybe several thousand to be sure, and we have a number of experiments, maybe several hundred experiments. When the value is above a median value, that number will be represented by a red spot. If it is below a median, it will be represented by a green spot, and in this case, black spots are values that are close to the median, so that you can see, for instance, in this condition, you see a bit of a red stripe. So in general, gene expression levels are higher in this condition than others. Because you can see patterns here, this is not the way the experiments are done, but these columns and rows are organized in terms of ways that maximize the similarity between the two rows. By arranging the rows, the genes, in an order, we're going to look for genes that have similar expression values. And the reason we do that is if we can find, and this will be clearer on the next slide, if we can find 10 or 20 genes that are doing the same thing at the same time in the same conditions, and we know what the function of one of those is, we will have a good guess as to what the other 19 genes are doing in terms of function. Similarly, we're going to look for experiments where thousands of genes have similar expression profiles. This might be a case where we're looking at lots of different cancers, and we might identify subtypes of cancers that molecularly have similar expression patterns. Those might be treated the same way. And here you see the patterns a little more clearly. In this case, only 1,500 genes are represented from a larger sample, and the expression has been looked at 529 samples, and you can see that samples which have similar profiles have been arranged to be near each other. Again, genes are in this column, and look, here's a set of perhaps 40 genes or so that are have high expression. Here's the legend for high expression versus low expression. High expression in these first 100 conditions, lower here, higher here, and again, the idea is to find genes that are behaving the same and also to find samples that are behaving the same. So how do we do this technique where we're going to mathematically compute which rows go near each other? Well, there are a lot of variations in the theme, but essentially we're looking for different similarity measures, and one commonly used similarity measure, which you've heard of before, are correlation coefficients. This is going to indicate how similar two things are. If we look at expression patterns for gene A and gene B in 6 different conditions, 1, 2, 3, etc., we can see that these genes are anti-correlated. The coefficient of correlation for these two genes would be -1 across these 6 conditions. These genes are not very similar. Here in the second example, we have two genes that are actually very well correlated. However, as they're represented here, they will not, that would not come across mathematically. So there's a couple things that we have to do to pre-process the data before we calculate coefficient of correlation. The first thing we're going to do is we're going to mean center. So we calculate the mean expression level of these genes individually. Those are indicated by the dotted lines, and we're going to center them. So we're going to subtract a constant, basically, in this instance, from the blue gene expression levels, so that it's superimposed right on the red level. But we also have a greater variance here, so we're going to variance normalize. Compute the variance for each of these two gene expressions, divide by that, so we're going to have essentially unit variance for a higher expression and lower expression values. Then when you compute the correlation coefficient, these would have correlation coefficients of essentially 1. And it's something your eye can pick out right away, but we have to prepare the data so that computers can actually calculate that. So these genes, being very similar, would likely be represented on 2 rows right next to each other in our large matrix, or our rearranged matrix, after the clustering analysis. So there are many different clustering techniques, and the one we will spend time talking about is one of the conceptually clearest. It's called hierarchical clustering, also referred to as agglomerative clustering. And this is a completely deterministic method, and what I mean by that is this is entirely based on the numbers. If you do the same analysis 2 times, you will get exactly the same result, because it's entirely based exactly on the numbers. There are other methods which have random processes, random steps, or optimization processes in which you will not necessarily get the same result if you do the analysis twice, although you would hope the 2 times you do it you'll get similar answers, but not exactly the same. Both approaches, I should say, have their advantages. Now let's say we have 100 genes by 50 conditions. So at the beginning of the analysis, and now we're going to talk about clustering the genes first. Each gene is a cluster, so we have 100 clusters for 100 genes. In each of our calculating steps, the 2 closest clusters, meaning the 2 clusters with the highest correlation coefficient, we will merge that into one cluster. And the values associated with one cluster, again, you can make lots of variations here, but let's just say it's the average of the 2 clusters which comprise it. And we'll repeat this step 99 times so that we'll only have 1 cluster left at the end. Once you cluster the genes, you could do exactly the same thing on the other axis in the columns with the experiments. In this example, we have 5 genes, A through E, and in the first round, we're going to calculate all the pairwise correlation coefficients, A with B, A with C, so on, D with E, C with D, B with E, all pairwise correlation coefficients. It turns out in this example, genes A and B have the highest correlation coefficient, and so we will merge a new entity. We'll have this new entity called AB with average values between gene A and gene B, and average values of expression across all the conditions. So now we have 4 items, A, B, C, D, and E. Then we will once again calculate all the pairwise correlation coefficients, AB with C, AB with D. We already know what D and E is, all the rest. And then in this analysis, in step 2, we see that D and E have the next closest correlation coefficient. And we will continue this, and in the third step, we'll see that this combined entity, gene DE, has a high correlation coefficient with gene C, and so on until we get to the final stage where we have a cluster that represents all the other genes. If we compare it to a gene expression profile, I've just tilted this on the side, but you can see these the shortest bars indicate those genes that have the highest correlation coefficient. There are a lot of genes here that have very high correlation coefficients with essentially median expression in these conditions, low expression in these intermediates, and all these genes have very similar profiles. And then you can see the farthest separation right here is between genes that are basically up versus genes that are basically down, and that represents this node right here. So now let's say you've completed that analysis and you've clustered the genes and you've clustered the conditions as indicated by the dendrogram here at the top. You can tell you've clustered by the genes. You see these stripes of similarly behaving genes. And then these authors in this particular paper have picked out 3 different regions which are blown up here. You can convince yourself of that. Here we have sort of a green, then a red patch, then green, green, red patch, then green. And you might say, okay, well, what do these similarly behaving genes do? You might sometimes have a clue. I see here genes that are involved with protein degradation. I see genes here that involved with protein creation, maybe some transcription factors. Maybe this is all proteins that are related in some sort of turnover process. And so you might then form hypotheses based on this sort of discovery science about why these genes might be behaving more similarly. But let's say you look at this next set of genes and you don't have a good idea of what they might do. Maybe you're not familiar with some of these genes. So what you'd like to do is submit this list of genes, and I'll work forward with this set of genes as an example. Submit them to a database. I personally don't know too much about these genes, but these databases have a lot of information stored about these. See if you can identify any patterns amongst these genes, and then tell me how likely it is that this is a real phenomenon versus a statistical fluke. So the way that people do this, or the basic approach, is something called enrichment analysis, and it's based specifically on a tool called the hypergeometric distribution. And I'm not particularly a statistical expert, but I know how to use the tools. And formally, this is the hypergeometric distribution describes the probability of k successes in n draws without replacement from a finite population of size n containing exactly k successes. So I've put that in terms of the genome, and here's the formula. Here are the variables defined, and if we say that the human genome has approximately 24,000 genes, so a finite population of size n is 24,000. And 875 genes have been identified in this database as playing a role in apoptosis. So k successes means 875 are labeled specifically as being apoptotic. Then if we were to look at a cluster that we had achieved through cluster analysis, and there were 17 genes in this cluster, and 6 of those we know play a role in apoptosis. So then k successes, which is 6 out of n draws, which would be 17. Now the question is, if I find 6 genes that are involved in apoptosis out of 17, is that significant, or can I expect that just by chance? Well, you could do that using this formula. This formula is embedded in Excel if you want, in a not super convenient way, but there are many online tools that will do this analysis for you. And then here I have the cluster of genes that we're interested in, and I'm going to use an online tool. And we will work together in class doing some enrichment analysis when we do the module on bioinformatics. But this is a very convenient online tool. We're going to use the functional analysis tool section, and then, although it's hard to see here, I can assure you that I've entered this list of genes in the data entry file, or you can upload a file. As background, I'm taking all the genes in the human genome, and I will do a functional analysis test. They will analyze this for functional analysis along many different considerations, but one of the ones that we're interested in is molecular function, which is characterized by the gene ontology. Again, we will deal with that in the lecture on computational biology, and I want to look at the chart that is related to biological process. I want to know what are the biological processes my list of genes are involved in. So if I click on this link, I will get this output, and we will blow this figure up. And here are the results blown up. And we have a number of cellular processes which a number of the genes in our list are involved in. And we look at the p-values, and initially we say, 0.007, that might be significant. But we'll get to that. And we're looking at the various processes that seem to be enriched in this sample, and well, we see the term homeostasis listed frequently, and so we start to develop hypotheses about what this group of genes might be doing. But here's a very important thing to know. Many times people will report p-values that seem significant, but one of the things you need to do is you have to account for a multiple hypothesis correction, which is to say that if you make many different hypotheses, each with 10% chance of being right, if you have 10 hypotheses, then just statistically on average, one might appear right. And so you have to do a correction to the p-values. In this case, there's some common ones, Benjamini-Hochberg or the Boniferoni corrections, and once you apply these corrections, you'll see that you have essentially a p-value of 0.76, which is not significant by any standard. So on the one hand, you wish that the list of genes that you were given were going to produce valuable insight. And in this case, well, although there are some suggestions, the corrected p-values are telling us that really there's even the databases are not aware of common functionality of the genes we submitted, which are statistically overrepresented in our set of genes. Another way of saying that is if I pick any 11 genes randomly out of the 24,000 in the human genome, I might find just as many playing roles in amyloid precursor protein metabolic processes or cellular ion homeostasis as the list which I've submitted for analysis. So anyway, that concludes our brief look into the analysis of gene expression data. Thank you for listening.